An Introduction to Polymerase Chain Reactions
by Caitlin Kang
and Michaela Nickerson.
PCR is the process by which a target strand of DNA is isolated and amplified. You start with a complete strand of DNA. After PCR, you will have many copies of that specific sequence.
There are 4 components of the PCR process. First is the template DNA, which has the desired sequence on it. Second is the primer DNA, which will mark the starting and ending points of the sequence. And third are the nucleotides, which are the A's, T's, C's, and G's that are used to make the replicated strands. And fourth is the DNA polymerase, which is a protein used for the process of putting together the nucleotides in the correct order. There are 3 steps to PCR. 1. Denaturing, 2. Annealing, and 3. Extending.
Denaturing is when the 2 strands of DNA split at a high temperature, usually around 94 degrees Celsius. Let's take a closer look at denaturing. In order for the 2 strands of DNA to separate, one must overcome the hydrogen bonds holding together nucleotides. In adenine and thymine, there are 2 hydrogen bonds holding them together. In cytosine and guanine, on the other hand, there are 3. At temperatures of around 94 degrees Celsius, there is enough energy to pull those 2 strands apart.
The next step in PCR is annealing, and this happens at around 64 degrees Celsius. The temperature lowers so that the nucleotides on the primer can form hydrogen bonds with the nucleotides on the DNA. The primer attaches to corresponding sequences on the DNA on either side of the target strand. It's important that the primers anneal on either side of the target strand in case we're looking at a repeating sequence. Take, for example, a strand that repeats ACG, ACG, and the corresponding strand would repeat TGC, TGC. Let's say the primer is set down at AC and GC, which would be convenient, of course, because that's what the entire target strand starts and ends with. However, there are then multiple places where the primer can anneal to, and the sequence could be cut up in many different places, and therefore not resulting in the entire desired strand. If the primers were to anneal on either side of the target strand, however, although we may result in some extra nucleotides, we will have the complete target strand.
The next step is extending. The DNA polymerase attaches itself to the primers in either end of the target sequence and extends the replicated strand by adding nucleotides as it moves along. This is made possible because the polymerase we use comes from TAC, which stands for Thermus aquaticus, a bacteria that lives in hot environments and therefore will not denature at the high temperature needed in PCR. Here's what it looks like.
Now that we have seen the 3 steps of PCR, let's look at the big picture. In order to result in completely replicated and isolated target strand, we must undergo the process 3 times. In stage 1, we start with the template DNA, which denatures, and then the primers anneal and the sequence extends. In stage 2, the replicated strands denature, primers anneal, and the sequence extends again. And then in stage 3, again, the replicated sequences denature, the primers anneal, and then the sequence extends. Only then do we result in completely isolated target strands.
By stage 4, we have 16 total copies of DNA, and we have 8 copies of the target strand. As the number of cycles increases, the total number of DNA copies increases exponentially, and the number of target strands eventually catches up and increases at a fast pace as well.
And that's all. Thank you for watching!
You needed more PCR in your life.